Yeah, it is a very interesting topic, the beta cell dysfunction and the de-differentiation. I think probably the story of diabetes begins with beta cell and ends with the beta cell. If you look at this, what causes the beta cell failure? There are different ways it can happen. Compensation by beta cell works only if the cells are robust. In susceptible beta cell, compensation fails. The defect creates weak link in the compensation process that promotes beta cell dysfunction, and hyperglycemia, once established, lead to glucotoxicity, promoted severe beta cell failure, and ultimately it lands up into the uncontrolled diabetes or hyperglycemia. The contribution of beta cell mass and function to the pathogenesis of type 1 and type 2 has been well depicted here, and the insulin resistance is both hand in hand with the type 2 diabetes. The insulin resistance and beta cell dysfunction are the core defect in type 2 diabetes, which has been very well established. The possible beta cell fates in type 2 diabetes and ways for recovery of the functional beta cell. Once the functional beta cell, there could be genetic predisposition and the cellular stress, which can lead either to beta cell death, which could be dysfunctional beta cell, or it can ultimately lead to de-differentiated beta cell, which finally goes into this kind of selection. The pathogenesis of type 1 diabetes, all of us know the development of type 1 diabetes is thought to be initiated by the presentation of beta cell peptides by antigen presenting cells, APCs. And these APCs bear these autoantigens migrate to the pancreatic lymph nodes where they interact with the autoreactive CD4 T lymphocytes, which in turn mediate activation of autoreactive CD8 T cells. These activated CD8 T cells return to the islets and lyse beta cell expressing immunogenic self-antigen on major histocompatibility complex class 1 surface molecules. It's a very intricate pathological cascade. The beta cell destruction is further exacerbated by release of pro-inflammatory cytokines and reactive oxygen species from innate immune cells, and these macrophages, natural killer cells and neutrophils brings about the beta cell apoptosis. These autoantibodies can be measured in circulation and are considered a defining biomarker for type 1 diabetes. At diagnosis, people with type 1 diabetes have reduced beta cell function compared with the healthy controls. With amelioration of hyperglycemia, these beta cells can have a partial recovery of insulin secretory function, leading to so-called honeymoon period after diagnosis with minimal or no exogenous insulin needed. The beta cell abnormalities might also contribute to type 1 diabetes pathogenesis, leading to what is called a beta cell suicide, beta cell hypoglycemia. Beta cell HLA class 1 overexpression is common in pancreatic sections from cadaveric donor with type 1 diabetes. Overexpression serves as a homing signal for cytokine T lymphocyte. Additionally, evidence also exists for increased beta cell endoplasmic reticulum stress linked with accelerated beta cell death. Endoplasmic reticulum stress in beta cell has also been associated with alteration in messenger RNA, splicing, and error in protein translation and folding. Resultant protein products have been proposed as a potential immunogenic neoantigens. Type 1 diabetes is characterized by a variety of abnormalities that involve both islet and the exocrine pancreas. Hallmark of type 1 diabetes is the loss of insulin-producing beta cells and immune infiltrates of islets. Type 1 diabetes mellitus is a chronic autoimmune disease with a strong inflammatory component. Inflammation might contribute to early induction and amplification of immune assault against pancreatic beta cells. At later stages, no stabilization and maintenance of insulitis. There may also inhibit or stimulate beta cell regeneration and might cause peripheral insulin resistance. So the important key points are innate immunity and inflammatory mediators, the activation of both endogenous and exogenous lesions or pattern recognition, amplification of insulinitis might depend on dialogue between immune cells and beta cells, After transition to adaptive immune response, inflammatory mediators such as cytokines might contribute to prolonged functional suppression, and some inflammatory mediators might promote, and that's all leads to a severe autoimmune phenomenon. Inflammatory process that might contribute to type 1 diabetes has already been talked about. Interaction of beta cells and immune cells lead to induction and amplification of insulinitis and transition from innate to adaptive immune response. During progression of type 1 diabetes, beta cell most likely die from a combination of necrosis and apoptosis. Very important statement. Methods to visualize beta cell inflammation and beta cell mass are being developed. Molecular techniques have been applied to identify beta cell killing in vivo. And basis for long-term loss of beta cell remains unclear. It is possible that ongoing beta cell destruction is intrinsic. This is proven beyond doubt that these abnormalities are well depicted and histologically in vivo analysis. For example, C-peptide, insulin and proinsulin studies have provided evidence for the persistence of insulin-producing beta cell in long-duration type 1 diabetes. There are persistent mechanisms by which beta cell apoptosis has been taking place, and viral infections in type 1 diabetes also hits the beta cell. When the pancreatic beta cells are subjected to autoimmune attack, killing the majority of beta cell population while the neighboring alpha cells and delta cells are spared. Destruction of beta cell manifests as a failure to produce insulin. Consequently, patient with type 1 remains insulin dependent for their lifetime. And that is very important. There are certain viruses like enteroviruses, Coxsackie viruses, which trigger these autoimmune process. Many candidate genes for type 1 such as MDAS5, PTPN2, and TYK2 regulate antiviral responses in both beta cell and immune system. There has been decrease from clinical onset of type 1, deterioration of glycemic control associated with different degree of insulin resistance. There has been a beta cell dysfunction and mass which gets totally to the root cause of autoimmune type 1 etiology of the diabetes. Pancreatic beta cells are extremely sensitive to the blood glucose concentration. Changes in its homeostasis influence beta cell function and population, which is proved beyond doubt. Factors which are associated with progressive reduction of beta cell function and mass are: one is a lipotoxicity, which is well established. Prolonged exposure to increased free fatty acid may have negative effect on beta cell function and lead to accumulation of toxic fatty acid metabolites in the islet cell. The autoimmunity and inflammation, markers of autoimmunity in phenotype 2, beta cell function, a possible role of immune system in the pathogenesis of type 2 diabetes. About 10% of individuals with type 2 diabetes, specific autoantibodies have been found out. Adipokines, hormone-like peptide on proteins released from adipose tissue. Leptin, pro-inflammatory cytokine, contributes to beta cell failure, may induce inflammatory reactions and apoptosis upon chronic exposure. Islet amyloid. Islet amyloid deposits in pancreatic islets have been considered a pathological character of type 2 diabetes, and its role in beta cell death has long been researched. Incretins, through binding on specific receptors on beta cells and increases of intracellular cyclic AMP and calcium, incretin stimulate the glucose-stimulated insulin secretion, improving both first and second phase response and restoring the biphasic profile. Because the stimulation of insulin synthesis occurs by upgradation of insulin transcription and translation and stabilization of insulin mRNA, beta cell secretory capacity and insulin stores are preserved. So there are multiple mechanisms which have been proposed for the beta cell dysfunction as depicted in picture. It is speculated that after a prolonged period of insulin hypersecretion, depletion of readily available insulin granules may occur, thereby reducing the insulin secretory response, may play a role in wider multifactorial setting of diabetes pathogenesis. Beta cell exhaustion The strongest support for exhaustion concept comes from studies examining the benefit of beta cell rest strategies. That supports the beta cell exhaustion theory. When inhibitors of insulin secretion such as digoxide or somatostatin are given to animals or humans with diabetes, paradoxical increase in beta cell function are observed following a period of rest. This is a very strong documentation of this beta cell exhaustion theory. Furthermore, human subjects with poorly controlled diabetes demonstrated dramatic improvement in beta cell function following a brief period of intensive glycemic control. Assessment of beta cell function in main pathogenic mechanism can be done, and changes in insulin sensitivity. In fact, the proven that between the insulin secretion and insulin sensitivity, there is a non-linear hyperbolic relationship, and product of two variables is constant, disposition index. Therefore, a correct evaluation of beta cell function requires quantification of the insulin secretion in relation to insulin sensitivity, evaluation of disposition index, which is very important. There are surrogate methods of evaluation of beta cell function: fasting insulin, fasting C-peptide, HOMA-B, HOMA-2B, fasting proinsulins, intravenous stimulation test, intravenous glucose tolerance test, hyperglycemic glucose clamp, graded glucose infusion, and arginine stimulation test. There are various methods: oral stimulation test, oral glucose tolerance test, and mixed meal tolerance test, insulogenic index. That is a way we can assess. Understanding of beta cellular in type 2 diabetes, renewed focus on beta cell differentiation. How this may be prevented or reversed, extent to which other islet cells can be induced to differentiate into beta cell. Recent therapeutic developments, possibility of restoring endogenous beta cell function, although long-term stability, safety, and efficacy of these approaches is not yet known. Beta cell dedifferentiation in the diabetes is important, but what is this? When we talk of beta cell dedifferentiation, what after all it is? Dedifferentiation state of a fully matured beta cell can change with natural reversible events and during the progression to death. Hyperglycemia of diabetes is thought to be major determinant causing beta cell to lose their differentiation, also termed dedifferentiation, which leads to the dysfunctional insulin secretion. This process is also called as glucose toxicity, which can be at least partially reversed by treatment of hyperglycemia. Mechanism of beta cell dedifferentiation in diabetes. Recent findings indicate that process involves coordinated and tightly controlled activation, repression of a specific transcription factor that drive transition from a stem cell to a mature beta cell during embryonic development and postnatal period until weaning. And this is proven beyond doubt the methods of beta cell dedifferentiation. Beta cell dedifferentiation characteristics were observed in mouse pancreatic insulinoma, beta cell exposed to high glucose for a long period, but massive cell that did not occur. A recent study showed that beta cells in type 2 diabetes patients lose their identity and cell mass, possibly even gaining features of other islet cell type, indicating that islet cell remodeling with dedifferentiation is the underlying cause of beta cell dysfunction during development of type 2 diabetes in human. Despite significant alteration in beta cell differentiation, phenotype, and decrease in the beta cell mass in human type 2 diabetes, apoptotic rate of beta cell remains relatively low. Growing evidence suggests that beta cell dedifferentiation may be a potential mechanism for loss of beta cell function and mass during development of type 2 diabetes. MicroRNAs potentially associated with beta cell dedifferentiation have been found, and these microRNAs are microRNA 7, 24, 30, 124, 184, and these multiple types of RNAs are there. So when we talk about beta cell dedifferentiation, there's a strong take-home message. Pivotal role of beta cell dysfunction in progression of diabetes, many factors that influence beta cell function altering natural course of disease. A correct assessment of beta cell function require concomitant evaluation of insulin sensitivity because two variables need to be judged in relation to each other. The clinical considerations are made regarding the therapeutic option, which impact on various factors or mechanisms that contribute to progressive loss of beta cell function and mass. Pancreatic beta cell dysfunction plays a vital and complex role in pathogenesis of type 2 diabetes. Ultimately, it is a feature of beta cells to respond to the insulin requirements posed by the prevailing level of insulin resistance that leads to the development of type 2 diabetes. Various genetic and environmental factors conspire together in this state of beta cell susceptibility and decompensation. Thank you very much. And I think that's a brief presentation all about the beta cell dedifferentiation. Thank you very much.